Hi, my name is Kathleen. In this video, I'll be teaching you about an important developmental process called boundary formation. We all start out as just a ball of pluripotent stem cells. From there, cell differentiation produces hundreds of diverse cell types. Simultaneously, boundary formation organizes these cells into interconnected but functionally distinct units. During vertebrate embryonic development, a process called segmentation organizes the bilaterally symmetrical anterior-posterior body axis. Segmentation relies on transient developmental structures called somites, shown here in the center as imaged with the scanning electron microscope. Somites give rise to several tissues, including skeletal muscle, cartilage, tendons, and skin. To explain somatogenesis, we'll be using the chick as a model organism. As highlighted in the bottom right of this life cycle diagram, somatogenesis occurs after gastrulation. During gastrulation, epiblast cells ingress at the primitive streak and give rise to the paraxial mesoderm. As Henson's node regresses posteriorly along the primitive streak, somites are produced in its wake from the paraxial mesoderm. What happens to the paraxial mesoderm to form a somite? Cells on the boundary of the somite undergo a mesenchymal to epithelial transition, in which the relatively loosely connected mesenchymal cells adhere tightly to form epithelium. This mesenchyme to epithelial transition is controlled by a clock and wavefront mechanism. The clock consists of periodically cycling genes that induce epithelialization. The wavefront is a gradually moving position where the presemitic mesoderm becomes responsive to the clock. Let's take a closer look at these two mechanisms, beginning with the wavefront. The wavefront is created via the expression of opposing molecular gradients in the presemitic mesoderm. Already produced somites at the anterior end of the embryo produce retinoic acid, while Henson's node at the posterior end produces FGF. The retinoic acid gradient modulates the extension of the FGF gradient. The determination wavefront, marked here with a red bar, is determined by a relatively low level of FGF signaling. As somatogenesis occurs and the embryo elongates, the wavefront advances posteriorly through the mesoderm, making cells become responsive to the segmentation clock, which consists of periodic gene expression that sweeps through the mesoderm from posterior to anterior. Those cycles are shown here in purple on the left side of each sample. This cyclic expression is achieved via negative feedback loops in the FGF signaling pathway. Specifically, Wnt3a and Notch activate proteins which inhibit their own expression. With each cycle of gene expression, a new pair of somites is specified in the mesoderm regions that have already experienced the determination wavefront. The combinatorial expression of different Hox genes determines the identity of each somite. A 1972 transplant experiment showed that somite identity is already acquired in the presemitic mesoderm stage. Thoracic presemitic mesoderm transplanted into the cervical region of a different embryo retained its original positional identity and developed into thoracic vertebrae, producing extra ribs, as shown in the diagram on the top right. Below the diagram are photos of the extra ribs from the original 1972 paper. Subsequent research revealed the axial identity of each region of the presemitic mesoderm is set long before somatogenesis by Hox gene activation during gastrulation. Now that we've discussed somatogenesis, let's turn our attention to another process that requires boundary formation: corticogenesis. Corticogenesis is the process by which the six-layered cortex develops and involves the formation of laminar boundaries. As seen in the diagram here, each cortical layer is distinguished by a unique combination of cell types and projections. Neuronal birth date is the main factor in determining laminar fate. The later in development a thymidine injection is performed, the more superficial in the cortex the thymidine-labeled neurons are found. Thus, neural progenitors give rise to progressively more superficial neurons over time. Because deep neurons are produced before superficial neurons, the cortex is said to develop in an inside-out fashion. Neurons are born in the ventricular zone of the neural tube, then migrate away radially from the ventricle. Early-born neurons migrate using somal translocation, in which a neuronal process attaches to the pia and pulls the cell body up. Once the cortex thickens, later-born neurons migrate using locomotion, in which they use their neuronal processes to adhere to a radial glia scaffold and climb up until they reach the marginal zone. The Cajal-Retzius cells in the marginal zone, or layer 1, secrete relin, a molecule that signals to locomoting neurons to detach from the radial glia scaffold and thus terminate their migration. Laminar fate becomes progressively restricted throughout corticogenesis. The laminar fate of neural progenitors was studied using heterochronic transplants. In subpanel A, neural progenitors from a younger mouse were transplanted into an older mouse. Those progenitors adopt a later fate in the superficial layers. However, as shown in subpanel B, progenitors from an older mouse transplanted into a younger mouse retain their late fate in the superficial layers, rather than adopting an early fate in the deeper layers. Several similarities and differences exist between these segmental and laminar boundary formation processes. Let's discuss 3 of them, beginning with a shared mechanism. In both processes, a mature structure secretes a molecule that affects boundary formation. In somatogenesis, the already formed somites secrete retinoic acid, which modulates the FGF gradient and thus the position of the determination wavefront. In corticogenesis, already migrated Cajal-Retzius neurons in layer 1 secrete relin, which terminates the migration of newly born neurons. Another similarity is the production of different segments and layers along a time axis. In somatogenesis, somites are formed along a time axis based on when they have experienced the determination wavefront, with anterior somites produced first and posterior somites produced last. In corticogenesis, layers are formed along a time axis as dictated by neuronal birth date, with deep layers produced first and superficial layers produced last. The 2 processes have different properties when it comes to fate restriction. In somatogenesis, fate is highly restricted. Due to Hox gene activation during gastrulation, the fate of the presemitic mesoderm is determined before it undergoes somatogenesis. Thus, the presemitic mesoderm retains its fate even when transplanted to a different location. Meanwhile, in corticogenesis, fate is progressively restricted over time. Thus, young progenitors can take on intermediate and late fates when transplanted into older embryos. That's it for this video. I hope you enjoyed learning about the important boundary formation processes underlying somatogenesis and corticogenesis.